and phosphothreonine, and there's little, if any, detectable phosphotyrosine. Okay. However, if you now look at VSARC transformed cells in which we've introduced the viral SARC gene, say we infect cells with Ralph sarcoma virus and allow a lot of production of the viral SARC gene, and then feed those cells radioactive phosphate, and now look at the amino acids that are phosphorylated in those cells, one sees that phosphotyrosine essentially takes over as the predominant amino acid that is phosphorylated. What does that suggest? Yes? A mutation? What kind of mutation? A mutation of phosphoserine and threonine into phosphotyrosine? Okay. Anyone else? Yeah? An exchange of amino acids caused by what? Yes. Which gene? Viral SARC. So all we're doing here is taking cells that now have expressed viral SARC and looking at the amino acids that are phosphorylated. And what we see is a switch from the normal serine and threonine to predominantly tyrosine. So tyrosine now becomes the predominant amino acid. Why? Yeah. Okay, so we're getting very close. So he's saying something about viral SARC is causing tyrosine to become a major amino acid phosphorylated in these cells. Yeah. Yes, beautiful. The viral SARC is a protein tyrosine kinase, meaning the specificity for SARC is tyrosine. Specificity for cyclic AMP-dependent kinase is serine and threonine. But the specificity for this new kinase, this growth control kinase that causes cell transformation, is tyrosine. And when we infect cells with the viral SARC, express the viral SARC gene, we get a hell of a lot of kinase activity that's directed at tyrosine. That doesn't mean, so maybe what's misleading about this is it doesn't mean that we've lost serine and threonine. It's just that at similar exposures, serine and threonine become minor relative to tyrosine. They're still there in the same amounts as before, but to get an exposure which looks reasonable, it's almost all phosphotyrosine relative to serine and threonine. So we've introduced a protein tyrosine kinase into cells that is responsible for cell transformation. This was a major finding, simple experiment, but a major finding for its implications, which are that there's a new class of protein kinase that recognizes tyrosines instead of serines and threonines, and that's responsible for uncontrolled growth as an oncogene. Okay? So clearly, we'd like to understand now what is the mechanism by which the normal cellular SARC gene folds. Okay. What is the kinase activity of the normal cellular SARC gene? And what is it about the viral SARC gene that differs from cellular SARC through mutagenesis that causes it to have such a dramatic effect in cells? So I should mention that if you repeat this experiment, introducing the cellular SARC gene, essentially overexpressing more of the cellular SARC gene in cells. Cells normally have cellular SARC. If you put more of it in, you don't see any significant change in the phosphoamino acids that are produced. What does that suggest about cellular Sark? We put normal cellular Sark into cells. We don't see this type of change of a dramatic increase in phosphotyrosine in cells. And cells, of course, have normal cellular Sark. Yes? Okay, it needs more to activate it. What does that mean? Forget about that it needs more, but if it needs something to activate it, it means that it's not very active. Okay, good. So it's not very active. It is not an active tyrosine kinase. So if we put cellular Sark into cells, we don't see much of a difference. It is not normally very active. So the difference between cellular Sark and viral Sark is the level of activity. One is an extremely active tyrosine protein kinase, and the other is inactive, relatively inactive, in the normal cellular form. Okay, so now let's look at normal cellular Sark, which I showed you the crystal structure of, with all the domains and how they fold on one another. And here's a cartoon of that. And we see that there are several domains that are very important to protein folding and functional activity. The first domain of interest is the C-terminal domain, the carboxy-terminal domain, which contains a tyrosine 527, which is normally phosphorylated in cells. So under unstimulated conditions, normal conditions with normal cell growth, this tyrosine is constitutively phosphorylated. It is always phosphorylated. And it has a high interaction with a domain called the Sark homology 2 domain. The SH2 domain, which is found on a number of proteins, first discovered in Sark, binds tightly to phosphotyrosine with the proper surrounding sequence. So in normal cells, SH2 domain is bound to the phosphotyrosine at the carboxy terminus. Another important folding occurs with what's called the SH3 domain, the Sark homology 3 domain, with an internal region that's proline rich. So we have folding, tight folding of the protein, such that the SH2 domain binds the phosphotyrosine on the C-terminal, and the SH3 domain binds to a proline-rich domain, and the rest of the protein is involved in the catalytic activity of the protein kinase. Okay, and the very active site contains another tyrosine that is well folded within this tightly folded, overall folded structure. In this normal configuration, this normal folding of this protein, the kinase activity of the catalytic domain is inactive. So if we take this protein and we introduce it into cells, you have an inactive kinase that remains inactive in normal cells, and it won't have any change in the level of phosphotyrosine. If we unfold this protein, the kinase activity will become activated. Okay. So let's look now at the structure of the viral Sark protein to try to understand what it is about the viral Sark protein that's different from the cellular Sark protein that causes it to be extremely active as a protein kinase. And that's seen very easily when we compare the structure, the sequence of cellular Sark to that of viral Sark. And what we see